Okay, so what is the properties of visual input information to the brain? And I'm going to ask a question during this video. Do you see blueness at the place where you're looking at? So the properties of the visual inputs to the brain are dependent on the structure of the eyes, which is totally understandable. And our visual phenomenology is shaped by the properties and structure of the eyes. And I'm going to introduce a couple of important structures here today. One is the retina, another is optic nerve, and two types of the photoreceptors, cones and rods, and the retinal ganglion cells that sends an output from eyes to the brain, and then the fovea. So let's start with our entry to the visual system, eye. So eye is consisted of many different kind of components, the pupil lens, iris, cornea, aqueous humor, and vitreous humor, and retina. That's the retina is a sheet of the things that are composed of many neurons, and then that sends an output into the brain through the optic nerve. And this is the electron micrography of the sheet of the retina slice, and that's taken from Christoph Koch's book. And you see photoreceptors, cones, and rods in here. And we'll see it at the later slides, but the retinal ganglion cells are located at the bottom of the thing here, around here. Okay, this is the ones that are sending the output. And then these things are the photoreceptor component. So this catches the photons and then sends the things are processed into this way and then sent to the retinal ganglion cells. So far, so good. But what's actually weird is that the light is coming from this way. So these intermediate structure is actually interfering with the catching of the light at the cones and rods, which I'm going to go into detail a bit in the next couple of slides. All right, so this is the bright field images of a living rod and the cone photoreceptors, and it's isolated. And then, as you can see, the rod looks like rod according to this kind of shape, outer segment. And the cones are called cones because this part looks like cone, triangular cone shape when it's isolated. And the cone comes with three different types, but from the looks, it doesn't seem to be different as far as I know. And the way it differs is this outer segment contains lots of pigment that is either sensitive to the long wavelengths or mid wavelengths or short wavelengths. And sometimes this is correspondingly called red or green or blue sensitive cones. Now, again, our eye structure, the part of the retina is now, I inverted the photograph I showed above because the light is coming from above and then it goes first, retinal ganglion cell layers and then eventually reaches to this, cones and rods at the end. Okay. And the interesting thing is that this seems to be not the kind of the default for all the animals. In the case of the octopus, actually they receive the light and then the cones and also rods like thing, photoreceptors are located just to observe, or absorb the light, optimally. And then that information is sent to other layers. And then like, our retinal ganglion cells send it to the brain. This is very different for the vertebrates. Vertebrates they do have this pigmented layer, photoreceptors always at the back. And then it's scattered, interfered by all these, ganglion cells and so on beforehand. It's very weird, and I don't really understand why this is the case, but that has been the way that the evolution has been working on. And the ganglion cells sends an output into the brain. And because of this, there is a consequence of the blind spot and so on. So it's quite interesting and intriguing why this is the case for us and then not for the octopus. And one more interesting thing about the octopus versus human or vertebrates that the octopus doesn't actually invert the image like us. Our lens invert the upside down image into upright at the level of retina, but they don't do that. So it's always curious to me why do we not see the world upside down? That's one thing. And also, how do octopuses experience the world? I really want to know this. And the way we can explain this in terms of consciousness research. All right. So leaving aside of this octopus interesting facts, trivia, the highest spatial resolution is achieved at the location called Fovea. So here, Fovea, as you can see, in terms of the schematics, it always has a dip in terms of the retinal structure, corresponding to reduced amount or lack of the associated cells. So retinal ganglion cells and other cells are actually avoiding to be present on the fovea where you are looking at, and that's the way the vertebrates actually compensate for the fact that we have this ganglion cells and other retinal related neurons to be present before the cones and rods. So they kind of open up the gate, and then the lights are coming to the cones as accurately as possible with minimal diffraction. So the fovea is defined in chapter 3 of the Christoph book as the central part of the fovea, about 1 degree of visual angle in size, the width of your thumb at arm's length, like this way. Okay, as is about 1.5 to 2 degree, and so it's a bit smaller than that, and that's the specialized area to ensure that vision there is as good as possible. Okay, but here's a weird fact. We do not have the blue cones for the central 0.33 degree. So if you put your finger, like this way, that's already 1.5, and 1/5 of that corresponds to roughly 0.33. And there, the claim here is that you don't have a blue cones or short wavelength cones. How can it be? But here's the image of the fovea in terms of a cone versus cone distribution. And I just want to make sure that you should never expect that our eyes or retinal cones are colored like this. This is just for the visualization, right? Everything doesn't have this type of color. Anyway, the left side is a cone distribution for the normal people who has 3 types of a cone: blue and also green and red sensitive cones. To be more precise, short, medium, and the long wavelength cones. And the right side is red-green colorblind people who has only 2 types of the cones, blue and green. And then in both cases, you see that the blue cone is absent here. Here, and also here. Okay. So let's do the experiment. Fixate the following image for 40 seconds, and then I'll count. And don't move your eyes and don't blink. Okay. So look at this center without moving your eyes for 40 seconds. Okay. I'll count. Ready, go. Don't move your eyes and don't close your eyes or minimize the blink if you can. Just focus and stare at the center of this blue circle. You may start to feel like the boundary of the circle becomes slightly blurry, but that's pretty normal. And the intensity of the blueness also starts to change probably, but that's also normal as well. And then 10 more seconds. Okay, pay attention to what you see at the fovea, okay? 3, 2, 1, 0. Okay. Don't move your eyes. And then observe. Okay, so if you are like me, then you probably saw some kind of, blue, yellowish kind of, after image against a white background. And in particular, the central part was slightly different kind of shade of the color. And I tend to see two rings, like, more intense ring and also slightly intense ring, but at least, you probably see one of them. And that is indicative of the lack of the color cone, the blue cone density. So that's the study shown in this, Magnussen's after image experiment. At least, you can experience there's something weird going on at the very center of the fovea. Okay, I hope that you experienced it. So this kind of the demonstration or investigation of your phenomenology using after image is one of the most important kind of technique in the consciousness research. All right, so summary, retina in the eye is a sheet-like structure, and from the retina, information is sent to the brain via lateral geniculate nucleus and other structures that I'm going to talk about in the following weeks. And while short wavelength sensitive cones are absent in the central fovea, we usually see blueness there. And that's really weird. Why? You need to think about it. And do we have other examples of unknown phenomenology induced by the structure of the eyes? Without answering the first question, I'll further ask questions, but this is quite normal in science. And that's the next kind of video, okay?